Welcome to the lecture series on vitamins and nutrition. Today, as a continuation from the previous session, we are going to look at one more vitamin, that's vitamin riboflavin, and today's content would be we are going to look into the structure of riboflavin, and we are also going to look into different aspects of riboflavin's function, its metabolic role, and we would start riboflavin with a historic note of when this particular vitamin was characterized to be used in therapy, and then we are going to systematically see its metabolic role in human physiology and what riboflavin is going to offer, and suppose if there is a deficiency for riboflavin, what all associated conditions that we actually connect with riboflavin. And yes, it's not the least, but we are also going to look into those sources where you find riboflavin as a dietary supplement, and also different kinds of functions that would be peculiar for riboflavin, and any deprivation of riboflavin, what are the deficiency conditions that would result from it. So, we are going to start with riboflavin. So, this is the structure of riboflavin, but if you look at the picture, so you find this yellow color filled liquid in the end of the microcentrifuge tube. I purposely put it up over there so that we will know this particular vitamin has a characteristic fluorescence. Can you see that yellow color, yellow greenish fluorescence? So, this is very special about riboflavin, and in fact riboflavin, apart from the clinical uses and the clinical applications what we are trying to connect with, that has also a lot of, I would say, applications in polymer chemistry. So, it can be even used as a kind of an initiator for polymerization reactions. So, that way riboflavin is basically a very interesting versatile molecule. Now, if you look at riboflavin, it comes in the order B2. So, as a water soluble vitamin like B1 and B2, B3, B6, then we have B9, B12, all series of B complex vitamins. It is one of the ingredient in the B complex vitamin. Very special thing is that about riboflavin, among all the water soluble, its solubility in water or polar solvents is much less compared to other vitamins. So, that is something special about vitamin. Probably we can associate this variation in solubility connected with the ring structure of riboflavin, or maybe that kind of a hydrophobic feature that they possess. So, you look over here, so the riboflavin basically is very essential in the utilization of carbohydrate, protein and fat. So, for any metabolic role, for any kind of macronutrients to be processed in our body, apart from the enzymes which break them down and release energy, you need B2 vitamin riboflavin to really help these vitamins. So, that is why it is one of the fundamental cofactors. So, you see there these two cofactors FAD and FMN. We are going to expand them shortly. It is like flavin adenine dinucleotide and flavin mononucleotide, two important cofactors. In fact, it is very much required for all flavoproteins. Okay, so, if you do not have FAD and FMN, that is going to be a big problem for our energy metabolism, and our body would not be in a position to effectively utilize them. So, if you look into the name riboflavin, it is a very interesting combination. So, you look at this particular name ribose, okay, and the flavin. So, flavin basically is a root word which means yellow. So, we saw just now the color yellow over here. So, that is why this particular vitamin, it is called the yellow vitamin or riboflavin, and this ribo is basically the sugar molecule ribose. When this is reduced, you have a ribitol. So, ribitol is basically a hydroxy form of the reduced form of ribose is ribitol, because ribose also has a ketone or a aldehyde functionality. In other words, we call it like carbonyl functionality. Most of the sugar and almost all sugar, by definition, you have a polyhydroxy compound with the carbonyl ring consisting of, I would say, the keto or aldehyde functionality. But if you look over here, the ribose where the aldehyde or the ketone functionality is being reduced to give you an all alcohol format, that's called ribitol. So since ribitol is part of the structure, it's called riboflavin. So if you could look into the structure of riboflavin, probably you will appreciate this better. I am going to explain that structure shortly. So this is the flavin, I would say, nucleus, and can you see over there this side chain hanging up, and this side chain attached to the nitrogen is basically the ribose or ribitol ring derived from the ribose. So you have a ribitol and you have a flavin, so that's why it's called riboflavin. Now we are going to look at the historic perspective of riboflavin. So you see that in the beginning I told you about this yellow green fluorescence. So this yellow green fluorescence was first reported officially by Alexander Winter Blyth in 1872. So he is the one who did this particular, I would say, association from the kind of pigment that you find from milk. So by virtue, you know, milk is a good source for riboflavin, especially you find that kind of an yellow tinge. You do have fat that's there, but in addition to that you also have riboflavin. Okay, so but although it has been reported that this particular compound is present in milk, but detailed structural characterizations have not started until 1930s. Okay, so the credit goes to three group, Kuhn, Georgi and Wagner. So three group of people, and here you have the Paul Georgi. So this team, they were doing lot of experiments on various extracts that can be used as supplements, especially for the treatment of deficiency diseases apart from macronutrients. We have already seen how the thiamine, the beriberi, anti-beriberi factor, was systematically evaluated in fowl species. Here what they have done is basically they have been feeding rat with thiamine supplemented diet. So you see there the growth failure of rats fed a thiamine supplemented diet. So what they did was they were trying to associate thiamine free extracts of yeast. All yeast and yeast related products are a very good source for these B complex minerals. So they were systematically doing a kind of, I would say, nutrient deprived extracts systematically, so that they want to know if you remove thiamine and then you feed the animal, what kind of effects the animal would show. So rat and mice had been very good mammalian models resembling the kind of diseases that human beings have. Most of the diseases can be replicated in mice and, I would say, rodent model, the rat model, and that's what they have done. So Georgi group actually had done this study and shown that there is a kind of a compound which is stable to heat, oxidation and acid, and it was sparingly soluble in water, but exposure to bright light, an alkali condition, destroyed it. Okay, so what they did was they found, I would say, more amount of riboflavin in bottled milk. So the moment you are going to expose this to a very high temperature, extremely high temperature or excessive exposure to sunlight, the vitamin component gets affected and its potency is lost. Okay, so here you find the structure. So this is little more detailed structure. So you see the riboflavin, and here you have the ribitol molecule present, and now we are going to look into the structure. So look here, this is D-ribitol. So what is this ring? So I have been telling you about this flavin ring. So technically this flavin ring is called in chemical terms as dimethyl isoalloxane ring. Dimethyl, this is isoalloxane ring. Okay, so if you are going to like chop up these two portions, in the middle you find something like a kind of a piperazine derivative, whereas this ring resembles more like, I would say, a pyrimidine ring. So a piperazine ring, pyrimidine ring fused together with one more phenyl ring, you get the isoalloxane ring. So this is the heterocyclic ring which forms the base for the flavin structure, and there in the middle, I would say the pi present ring, one of the nitrogen provides attachment for the ribitol nucleus. Okay, so ribitol is basically the reduced D-ribose that gives you, see, otherwise you are supposed to have, I would say, a keto group over here or an aldehyde group, but now you find all the groups are reduced, so it's a kind of a alcohol, I would say polyhydroxy alcohol. So this is the structure of riboflavin. Now this very, I would say, structure provides the way for fluorescence. In fact, acridin and many other dyes which show fluorescence have a structure resembling isoalloxazine, although it's like quite different, but this kind of a ring structure allows for fluorescence effect to be seen. Now if you are going to look at those two important forms, so riboflavin is basically the source for making flavin mononucleotide, as we call it as FMN and FAD, flavin adenine dinucleotide. Okay, so what we do or what we understand from this, they are prosthetic groups of oxidoreductase enzymes. In other words, we call them as flavoproteins. Those flavoproteins which has oxidoreductase activity. So what do we do? How we going to like really look at them? So we look them at a kind of, I would say, a metalloflavoproteins. Okay, you do have iron, you do have molybdenum as additional metals that would make the enzyme more functional. So you have this name, what we call it as metalloflavoproteins. Okay, so now you look at this FMN and FAD, they are required for a wide variety of oxidative enzymes, and they remain bound to the enzymes during the oxidation reduction reactions. Okay, so you look at this, you can consider the flavins, they act like a kind of an oxidizing agent, and because of their ability to accept a pair of hydrogen atoms. Okay, so you find them, their fundamental role would be revealed when you are going to look at them as part of FMN and FAD, flavin mononucleotide and flavin adenine dinucleotide. So wherever you require FMN and FAD as part of the metabolic enzyme cluster, you have to consider that would have been formed only with the help of riboflavin. Okay, now you look at this derivative. Okay, so you have flavin mononucleotide. So how do you really look at these two derivatives? So what is exactly this FMN and FAD? So these are all phosphorylated derivatives of riboflavin. Okay, so you find to the ribitol alcoholic group in position 5. So we saw this is the alcoholic group. In the position 5, you keep adding, I would say, phosphate groups, and the source for phosphate group is basically ATP. So if you are going to have a phosphate group with one phosphate molecule added from ATP, and that's, I would say, we get flavin mononucleotide. If you are going to consecutively add one more phosphate group, so that end of the day you have two phosphate groups added, you call it flavin adenine nucleotide. Okay, so these are all, I would say, linked through the phosphate components attached to the flavin ring. Okay, so these are two coenzymes of this vitamin. So this vitamin forms the raw material to form FMN and FAD. Now you look at this, here you find how this reaction happens, so that they act like a kind of binding the proton, you see over there, and the reaction being hypothesized this way. So you get this point. Look here, here you have the phosphate rings forming the chain, and here you have the flavin molecule. Now this is linked with the adenine. Okay, so you get this point. So adenine will be the nucleo component, I would say, the nucleic acid base. Fine, and here this is attached to, I would say, the riboflavin, and both are linked by the phosphate groups. since you have two molecules, we can call it like pyrophosphate groups. So this one links the actual riboflavin with the adenine molecule. So that's how you say flavin adenine mononucleotide. So if you are going to have a single phosphate group, and if you are going to have two phosphate group, you call it FAD, dinucleotide. Okay. So look at the structure. Here we have, there is this particular enzyme, the riboflavin, where you have FMN phosphatase, okay, the flavokinase. So how you have a kinase and a phosphatase doing the job. So when you have a flavokinase, you get FMN, okay, and you have one more, I would say, phosphorylation happening. So that adds one more phosphate group and you get FAD. So you see there, this is the structure of FMN. So you have only one phosphate group. If you are going to add one more phosphate group to this, you get FAD, flavin adenine dinucleotide. So this is how you write the structure. So here you find riboflavin and ATP being the source, you have flavokinase, so you get FMN, and this ATP transferring the phosphate group becomes ADP. So now you look at flavin mononucleotide FMN being acted upon by one more ATP molecule, okay, and again you have one more pyrophosphorylase, flavin nucleotide pyrophosphorylase in the presence of magnesium gives you flavin adenine dinucleotide, and here you have the inorganic phosphate generated. So this is the kind of activation that you get for FMN and FAD formation. We are going to look at the source. So you see there, milk is a wonderful source, fish is a source as far as the, I would say, animal protein source is concerned. Then you have green leafy vegetables, then you have pods, then you have cereals, wherever you have this yellow tinge, okay. So you can to some extent consider vitamin B2 presence, and then you have in the animal products beef liver, I mean liver for that matter. Liver is an excellent source for storage of many vitamins, and B2 is one of the vitamins being stored in the liver. So you look at this structure, here we have the phosphate group added. So you see initially riboflavin being acted upon by flavokinase, so that you get a phosphorylated group, then FAD synthase comes over there, and here you have the adenine group added over here. So you have one more phosphate group is added, and then in addition to that you have, I would say, FAD synthase producing FAD, adenine dinucleotide, okay. So that's very interesting to look at how the activation happens. So now we are going to look at what would be the role of riboflavin. I think every one of us sometime or the other would have had or shown symptoms of riboflavin deficiency. You may wonder how come that is possible, because whenever you look at riboflavin, we are like so used to this particular vitamin, and the demand for riboflavin, and along with riboflavin there is one more vitamin, the folic acid, both have always been a vitamin in demand. And since we are dependent on, I would say, vegetables and milk as a source, we might need more doses of riboflavin, especially when our body undergoes lots of physiological stress. One of the most common symptoms, although we are going to deal about symptoms later in detail, now it will be high time I tell you about the symptoms, so that it would be much easy for you to register and associate this vitamin and remember many facts about this vitamin in alignment with the deficiency. So what we call it like chelosis, especially chelosis is angle of the lips where it becomes like white. So that happens when there is a sustained, I would say, deprivation of riboflavin for a very long period of time. So such kind of deprivation for a very long period of time produces lots of epithelial damage and exposes the inflamed epithelial layer, which forms that whitish appearance In addition to that, we will also see symptoms like, I would say, mouth ulcers or mouth sores, again one of the most common symptoms. So chelosis accompanied by mouth sores is a characteristic sign or symptom of riboflavin deficiency. Okay, so you see there, it helps in the overall maintenance of epithelial tissues and fast growing cells. You see their skin, hair, eyes, nervous system, everything. Wherever you require the normal maintenance of epithelial cells, you always look at riboflavin. Not only that, any mucosal membrane in the gastrointestinal tract also needs a regular intake of riboflavin, because riboflavin helps in being constantly replenished whenever any damage happens on the surface of the gastrointestinal tract. See, mucous membrane will be always eroded because gastrointestinal tract is being constantly exposed, okay, to erosion and friction. The food we consume, the kind of metabolism happening, the peristaltic movement. So there is a bound damage to mucosal membrane, which is part of the normal process of digestion, because this would be constantly replaced. Without mucous membrane being replaced in the gastrointestinal tract, it's going to be very difficult, and for this replacement and for maintenance you require riboflavin. Okay, not only that, whenever there is any injury, whenever body is trying to repair itself, it also takes help from riboflavin, because riboflavin is the one which basically handles how you would provide sufficient nutrients for the restoration of membranes. So you see that also RBC membrane, okay, you do have problems with, I would say, frequent degradation of RBC membrane, and this is essentially being supplied by riboflavin for its restoration. Not only that, even in cases of migraine, although the mechanism is not yet known, but there are reports which suggest that migraine kind of conditions respond to riboflavin, or riboflavin deficiency can give rise migraine like symptoms, I mean a constant headache. Now if you look at very closely, any kind of headache or a constant headache would be the result of vasodilatation. So since there is vasculature assigned, and the mucous membrane being affected in the vasculature could also be I would say a reason behind migraine like symptoms. So that's one of the reason why a dietary intake or supplementation of riboflavin would try to stabilize the membranes of the vasculature and thereby even provide for the treatment of migraine. Now we are going to look into little more in the detail where exactly this mechanism happens. Okay, so you see what we call it as the redox potential. Okay, so they play a wide variety in the intermediate metabolism or intermediary metabolism. What do you mean by that? So when there is a series of metabolic reactions that's happening in the metabolism of macronutrients like carbohydrates and proteins and fats, you do have to count on redox potential of flavoproteins. Okay, so you see there lots of roles that they play. So in the electron transport plane, you have the flavoproteins contributing for a normal conduct of electron transport chain. Then for decarboxylation of pyruvic acid and alpha ketoglutaric acid, you require FAD as a source. Then you have, there is this particular enzyme, what we call it as fatty acyl CoA dehydrogenase. So this particular enzyme also needs FAD for its fatty acid oxidation. So you also find that FAD is a key component for the production of pyridoxic acid from pyridoxal. So pyridoxic acid is basically the active ingredient of B6, but you have to have a precursor, the pyridoxal, and for this production you require FAD. So each of it, as you know it, has some specific role to play. So you look here, FAD is required to convert vitamin A to retinoic acid. So without this, the function or role of vitamin A also becomes affected. So you see that riboflavin playing a major role in the retinal. In fact, we are going to see later how riboflavin plays a role in the treatment of cataract. So whenever there is cataract, thinning of membrane, a constant supply of FAD would help in the healing and restoration process. And here we find the retinal being converted to retinoic acid, and this restoration is also very essential in terms of the normal visual cycle. And you see over there, FAD is required for conversion of one amino acid to the other version. So tryptophan to niacin, okay. So required to convert tryptophan to niacin. Tryptophan is supposed to be an amino acid and also a kind of a neurotransmitter, okay, but you are using tryptophan as a source for niacin. So niacin is vitamin B3, which we are going to see elaborately in the later sessions. And you also see that there is this versatile tripeptide, what we call it as glutathione, which is a very interesting tripeptide, okay, and normally given by this notation GSSG, where SS refers to the thiol molecule, which can be reversibly reduced or oxidized. So glutathione, you write it like GSH when both the sulfur groups are, I would say, oxidized, means losing of hydrogen, they become GSSG. Okay, so this kind of effect, that glutathione mediated functions, which in turn is dependent on riboflavin, will tell you its important role as to where all FAD is needed. So FAD can be directly used in the synthesis of these amino acids and also this particular vitamin, and especially this glutathione. Where do you find glutathione? Glutathione is abundant in the liver, and it's regularly used in many kind of conjugation reactions. So how they are replenished? Only through this pathway, okay. Now you see there glutathione oxidation reduction, basically the redox cycle. So you see the reduced glutathione GSH, okay. So glutathione peroxidase in the presence of selenium. Now glutathione's ability to scavenge free radicals, especially hydrogen peroxide, okay. So this is the cycle for which normally we associate glutathione. So glutathione peroxidase in the presence of, I would say, the reduced glutathione becomes very functional. Then you have the oxidized glutathione. So reduced glutathione and oxidized glutathione are produced in a cyclic fashion. So the reduced one gets oxidized, and this happens with the presence of glutathione peroxidase, okay. Then there is glutathione reductase, which basically uses riboflavin and again I would say kickstarts the process. So this goes on and on and on. So for one molecule of hydrogen peroxide is reduced to 2 molecules of water. So hydrogen peroxide is a very potent, I would say, free radical donor, and this can cause membrane damage. Apart from the external sources of hydrogen peroxide, even in our normal metabolism you would find such free radicals are being released, okay. So these free radicals are being scavenged effectively by glutathione, okay, and then this in turn is being catalyzed by the seleno enzyme. We are winding up. I am going to give you a quick review of what we have done today. So we started with the historic note on riboflavin, the structure of riboflavin and the function of riboflavin. I have elaborated its metabolic role. We are going to have some questions. What is the structure of riboflavin? What is the structural components, and what is the way by which riboflavin gets activated in our human metabolic process? Then you can mention what are the rich sources of riboflavin, both plant sources and animal sources of food as dietary supplement. Then mention the metabolic role of riboflavin, how riboflavin contributes in the oxidoreductase enzyme cycle. Thank you for your attention.